So my great pleasure to introduce you to Gaia Navarino. It's a long-time friend. We know each other for, I think we overlap a little bit when you were here with Joe. But Gaia is a professor, and she runs like a lab at the Institute of Science and Technology in Austria. I visited there a couple years ago, and it's a beautiful setting. It's a great institute, great place to do science. It seems like everybody's interested in really fundamental, basic science in there. I was quite impressed by the settings and the people that I met over there. So I'm really glad that she found like a good place to work there. Before moving to Austria, she worked as a research fellow here at UCSD with Joe Gleason, and I think that's when we met, but also at the Max Dilbruck Center for Molecular Medicine and the Center for the Molecular Neurobiology in Hamburg. So she focused on understanding early stages of autism, or the interplay between genes mutated or affecting autism and neurodevelopment. And she uses a series of stem cells as well as animal models, and I believe she's going to be talking about some of these. The work that really impresses me the most, and I think was still in partnership with Joe, when they showed that there is a gene mutated in autism that actually leads to a specific pathway that could be treated with supplements. So to me, that was a game changer. I said, wow, I mean, it means that some of these conditions might be revertible if you know the pathway, if you know what to do later on. So it kind of forces me to go more into the genetics of autism, and this is part of my lab. So huge inspiration coming from Gaia and Joe in this aspect. So without further ado, thank you so much, Gaia, for accepting, for being here, and we look forward to your talk.
So thank you very much for the introduction, and of course for the invitation. It's amazing to be here. I actually remember San Diego a little bit more sunny, but I'm still really enjoying it. I mean, it's really a pleasure, and I'm very much looking forward to have conversations with you guys, both here and later during the day. So as Alison was introducing, so when in my title say neurodevelopmental disorders, I mostly say autism spectrum disorders. So just very briefly, it's a group of disorders that are very heterogeneous and associated with those social problems, like including language issues, and as well as the presence of repetitive and stereotyped behaviors. As we all know, those issues, they often go together with a number of other problems, including intellectual disability. Motor problems is actually something that we are becoming very interested about, because they're very, very common among autistic kids, and we really try to understand what are the relationship between the other features, core symptoms of autism, but also anxiety and epilepsy and so on and so forth. Now, for many years, we really knew just the very bottom of the iceberg, in the sense, or the very tip, sorry, of the iceberg, that we are associated always mutations with on autism spectrum disorders with genes encoding for proteins that are relevant to synaptic proteins. The reality came after, people like that sitting also here, like Jonathan, Joe, and many others, have done a lot of more genetic analysis of those conditions, and altogether found that eventually a number of other genes encoding for proteins that belong to a number of other functions are in fact involved and associated with those this group of conditions. And so now the question is in general, how we are bringing all of it together, and are they in fact those groups of genes and proteins connected with each other? And what I'm going to talk about today, I'm going to focus specifically on this class of genes that are really becoming one of the top class of genes associated with autism spectrum disorders, that are transcriptional regulators and epigenetic factors. More of it is what I really would like to, and we are really try to understand, is how we can connect this group of genes directly with what we know is happening in autism spectrum disorder, with the synaptic dysfunctions that we find in this group of disorder. And so what I'm going to show, if you want, is something about genomic and synaptic plasticity in autism spectrum disorder. And the whole work, in fact, started already a few years ago when we started working on a gene that some of you also are working on, that is SETD5. So together with some geneticists back then in between 2014 and 2015, when I moved to Austria, we found, and also in parallel to others, that mutations in SETD5 are a cause of intellectual disability, speech delay, short stature, and strabismus. So, and the number of patients that we identified, we and others identified, was quite large. So we know that SETD5 is actually one of the top high-risk genes. So SETD5, as depicted here, was associated back then to, so it belongs to a class of genes that is called histone modifiers, or so in this case, histone methyltransferases. And so what was supposed to do at that, so what we knew at that point, it was that this gene was involved eventually in transferring a methyl group to a certain histone in order to regulate gene transcription. And I'm not having the time to go through the entire data that we acquired back then, but basically what we found, and again, this was very rewarding to see that other people found very similar things at the same time or a little later. But what we found is that, in fact, SETD5 is not an histone methyltransferases. And we hope to have a cryo-EM structure of SETD5 with its complex very, very soon. We are working on that. But what we found is that SETD5 in fact interacts with two different complexes. One is this PAF1 complex, that is a complex needed to regulate the posing of the RNA polymerase, so to regulate transcription, and with the complex that is related with histone deacetylase, which of course is then directly modifying histones. And so, to make a very long story short, what we found is that in the absence of SETD5, the number of basically PAF1 complex associated with the transcription starting sites, specifically on neuronal genes, is larger, is much higher than in wild type. And so, as you will see, basically, as a consequence, those genes, those transcription starting sites, are much more prone to transcription. And when I say prone, I really mean that it's not that those genes are necessarily transcribed more all the time, but they are just much more prone eventually to be transcribed. And so the way I see in a way is like this picture here where you have, swimmers that are ready to go, and eventually someone is just starting a bit earlier than the others. So phenotypically, when we looked at the mouse lacking SETD5, one copy of SETD5, so this is what happened in patients. Patients have de novo mutations, so they are haploinsufficient, so they lack just one copy. What we found is that something that is relatively atypical, namely is that the brain size of those animals is completely normal, while the body size is quite smaller. So which was already a hint in saying that there is some sort of preference for brain-related or nervous-related type of cells over others. But then there was another phenotype. I love this one because it looks like such a, not very relevant to us when you're studying brain disorders. So one of our animal caretaker came to us and said, look, a lot of those animals have this white belly spot. And I mean, I promise at the beginning we were like, we're studying the brain, so who cares about the white belly spot? But again, I really like, because I think this is a beautiful example about how observing things can really teach us a lot, and about developmental biology. Maybe some of you know about that, but so the color of the hairs, of course, are coming from melanocytes in the hairs. And it happens that the melanocytes are deriving from melanoblasts, and melanoblasts are unequally distributed in the body very early, I mean also later, but very unequally distributed across the body, and being the belly, the part that has the fewest number of those cells. So whenever there is, and another piece of information is that those cells derive directly from the neural crest. So basically, whenever there is a neural crest, a differentiation issue, basically this is one of the places where you're going to see. So what we're going to see here is really specifically a neural crest differentiation issue. So and practically what happens is that the melanoblasts here are depleted, and so the very first part where you see it is in the belly. I just like to mention it because it's a beautiful example about how both animal caretakers are important, how our heroes, and plus how we can learn. So back then, what we were interested in understanding is still because we knew that this gene was relevant to regulated gene transcription, we went back and asked what happens at the transcriptional level in those animals. And so we looked very early on, actually, when mice were about embryonic day stage 9.5, because we thought that already because of this white belly spot, something should happen very early. We performed RNA sequencing of the wild type and mutant animals. And what we found is that, what I depict here, is that already at this stage, there was really a sort of bias of expression for genes. So here, whatever is in red, it means upregulated, blue means downregulated. And I love this picture because it almost resembles the embryo that is on the side. And as you can see, basically, anything that is related to the head and to the brain formation, not anything, but a lot of genes related to the head formation and to the central nervous system were upregulated, while a lot of genes related to other areas, including the neural crest, the skeleton, and so on, were all downregulated. So I really love the way it looks in this picture. Now, of course, this is we go almost like in a semantic, if you want. It's like, is what we are seeing here a phenotype of the fact that basically the embryo at that stage already has this small body size and a bigger brain, and so that's what we are seeing. I say it's almost semantic because if this happens, it must be because of gene expression, but still, we had this question in mind. And so what we decided to do back then is to generating human embryonic stem cells carrying either heterozygous mutation in SETD5 or wild type, and we did a lot of different things to have controls, and then again performing RNA sequencing there. And what we found is that already at this stage, what you can see in this heat map here, the two mutant lines that we generated show a upregulation of gene related to the nervous system development. So what we think is that, in fact, already at that stage, there is a bias because of SETD5 missing, and because SETD5 is usually regulating transcription at the transcription starting sites of neuronal genes. When this is not there, those genes are kind of more prone to be transcribed. And so that's what leads eventually to this preference of differentiation of nervous system relevant cells. So this is one aspect of the SETD5 work, but then of course we were still interested in the behavior of those animals to really go back to what autism spectrum disorders core features look like. And so we did a series, really battery of behavioral tests there, but one that I want to show, because it's relevant to the rest of the talk, is the context fear conditioning. And so I'm sure many of you are familiar with that, but just briefly, what we are doing is placing an animal in a new context that is characterized by certain visual and olfactory cues. And after about a minute of habituation, we deliver to the animal a few current shocks to associate this context with a bad experience. And so then we bring back the animal in the home cage, and 24 hours later we expose the animal again to the same context where they received the bad experience, and we measure the percentage, the amount of time that they spent basically freezing. And this measure is a measure of how well they remember that in that specific context they received that bad experience. And there was a bit surprising to see that SATD5 animals, they in fact freeze more than the wild-type animals, kind of hinting that in fact they remember better, not worse, than the wild-type, which is something that we did not expect. But what we also did is then we left those animals in the same context for about 7 more minutes, and this time we did not deliver any shock. So at this point, the animals should either form a new memory, it's not clear yet, either form a new memory or either delete the previous memory that was a bad context, and so they should be more relaxed next time that they are in there. And this is exactly what happened in the wild-type animals, but this is something that does not happen, in fact, in the SATD5. So we kind of conclude from that although those animals are very good in learning eventually, they are becoming inflexible to change, and inflexibility is indeed something that is very relevant to autism spectrum disorders. Back then, we also did the electrophysiology correlate of that is the long-term potentiation recording. So we stimulate the Schaffer's collaterals of those animals and record them from their CA1. And as you can see here, basically the synapses of the hippocampus in the mutant animals, they are much more longer staying potentiated than the wild type that here is indicated in black. And here you have basically all the quantification over the course of many hours. So we recorded up to 6 hours, in fact. And we see that these basically synaptic strands stay very stable over time. So practically, in a way, what here is this work was telling us is something eventually not very surprising, but eventually often overlooked, I would say. So whenever we are thinking about synapses or synaptic related disorders, we are really thinking about the synapses, but we are not thinking about what happens at the same time in the nucleus. So here the idea is that eventually there is a lot of communication between the nucleus and the synaptic and synapses to the extent that many years ago, in fact, someone was mentioning, kind of coming up with the term of genomic, I think, potential, so sort of to say like action potential, there should be also a nuclear potential somehow. And so that's what we wanted to follow up in the context of SATD5. And so what we did is repeating the behavioral analysis the behavioral training that we did before. So basically putting the animal, SATD5 and control animals in a context where we deliver current shocks. But then this time, instead of performing the behavioral task, what we did is obtaining the hippocampi about 1 hour and 3 hours after the training, and analyze them by RNA sequencing. And in this specific part, what we did is that, so when we looked at steady state, I should say, there was not major difference. So we could really not detect much difference between the genotypes. And so what we decided to do is really to cluster those genes or those transcripts that change upon the training of the animal in clusters based on their dynamic over time. So for instance, there are genes that eventually, like this one, eventually that it goes up 1 hour after the training. So this is home cage, 1 hour after the training goes up, and 3 hours after the training it goes down. And then others that have completely different behavior. So we basically cluster all the transcript that we obtain by RNA sequencing of those hippocampi after the training, 1 hour after and 3 hours after, based on their trajectory over time. And that's what we obtained. So we obtained many clusters of genes with different type of behaviors. And so, for instance, here you can see that this class of genes or transcripts in wild type, they are increasing 1 hour after the training and then decreasing 3 hours after. Those are, for example, the early, so immediate activated genes like CFOS, for instance. Those are very well-known responses. And then you can see that, so in yellow you always have the haploinsufficient mouse, and you can see that in many cases the mutant did not really, like in those cases here, did not really behave differently than in control. But then you also have a lot of clusters where, in fact, there are transcripts that have a completely different trajectories when one copy of SETD5 is missing. And so what we did next is asking what are the pathways that are enriched in those specific clusters, and we found that those 2 clusters that show this sort of persistent expression of certain gene were actually the most enriched for Go terms. So the others were also enriched, but so the p-value was a little bit less. But so we were looking at those that really were show a very high enrichment, and we looked what were containing. And indeed, what we found in those were containing really nicely, mostly postsynaptic density-related proteins, and among them we found many different ones that were already are associated with autism. So those that I depicted here in yellow and red, that were associated with other autism spectrum disorders. So practically, what we found here is that, so on one side, that SETD5 is associated with intellectual disability, ASD features, short stature, strabismus, etc. But then by doing some functional work, we came to the conclusion that eventually SETD5 is not just needed early, eventually during development, but is also needed to kind of establish this communication between the nucleus and the synapses to regulate the protein, the transcript levels upon during plasticity. So what we asked then next, it was like, is this something that is very specific to SETD5, or is this something that is instead we could generalize to other genes that are associated with autism spectrum disorder? And this is a general feature. And one of the reasons why we were really interested in that is that we thought, I mean, Alison before mentioned that when I was here in San Diego, one of the things that we found is that eventually in some cases we could rescue some form of autism spectrum disorder. And so one of the thought here was like, if synaptic plasticity and not just synaptic development is eventually relevant in those disorders and is regulated throughout life, this is maybe something that we can really target as a treatment later on also. And so what we decided to do is to go very systematic. And so we decided to include 3 more genes in our study. So those are H1L, KMT5B, and KDM6B. They're all top ASD associated genes, as well as epigenetic or histone modifiers. And so we really decided to go very systematic in understanding whether the mutations and haploinsufficiency of those genes was related, in fact, with as well with synaptic plasticity issues and how the two eventually would communicate. And here also we decided to make a step forward, not just looking at one form of synaptic plasticity. So what I introduced basically with the SETD5 work is the Hebbian plasticity, that is this form of synaptic plasticity that is related to learning. And so this is a form of plasticity that is specific to single synapses in a neuron. So that single synapses that are stimulated. And so what we decide, because I mean, of course, learning can be an issue in patients with autism spectrum disorder, but is not the only one and not one of the core one. And so what we decided now is also to include another form of plasticity, to study another form of plasticity that is called homeostatic plasticity. So this is a form of plasticity that instead of being just associated with one synapses, is really relevant to the entire, all synapses in one neuron. And the way I see this, and I like to explain it, is that is the way that neurons have to sort of reset their synapses to keep them in a certain dynamic range. So practically, the synapse needs always to do this range where it can go up, LTP, or go down LTD. But then if it gets to a point where they're saturated because they either, for instance, too many receptors or too few, you cannot go any more up and down. And so you need to reset the synapses to a certain range. Now, the important is that by happening this, involving all synapses in a neuron, the ratio, so the relation between the different synapses in a neuron always stays the same, because you're basically scaling all up or scaling all down. But then you put them in a range where this is more functional. And then, of course, this form of synaptic plasticity, it came to be very relevant for a number of different behaviors. So what we decided to do is to very, very systematically address this problem. So we generated other 3 animal models. So H1L haploinsufficient models, KDM6B haploinsufficient, KMT5B haploinsufficient model. We also have an inducible reversible system I will show to you later. And there, we decided to systematically study synaptic plasticity, so have both Hebbian plasticity and homeostatic plasticity, using both so electrophysiology, behavior, and as well as molecular analysis. Now, because one of the issues we often have when we study animals is that, and studying, human disorders in animals is like how relevant are those animals for the human disorder. What we also decided to do is for the same genes to generate organoids with starting off from isogenic cell line, so one cell line, using CRISPR-Cas, introducing a loss of function mutation in one of the allele of all those genes. And then in this case, we study homeostatic plasticity by electrophysiology. We cannot do behavior, of course, but and then molecular work using those organoids. So that's what I'm going to do. So this is all unpublished still, and I'm going to guide you through some of those results. So let's start from the behavior, and I'm not going to show you the entire data set. Some is still on, so we are about just analyzing it, but also it will be just redundant in a way. So let me start by showing you the behavior of 2 of those animals that are H1L and KDM6B in the context sphere conditioning. So this is the same paradigm I've shown to you before with SAT35. We are exposing the animal in a context where they receive an adverse experience, and then we are replacing those animals in the same context 24 hours later. And what we found is that during the training phase, the animals, so both genotypes, they are kind of able, so we are measuring always the learning with freezing, the amount of freezing of those animals. So during the training, we see that those animals, here in red and in the lila, they can really learn. So we can see that they're increasing their freezing as we start delivering some current shocks. However, when we measured their freezing about 24 hours later, what we found is that in this case, contrary to SAT35, in this case, the animals are indeed learning less. So they're freezing less, significantly less. And here I just put some one sex, but we're doing all of this also with males and females. So it's a lot of more layers of complexity. But what we are seeing is very similar. So we don't see major sex differences. But definitely having just one copy of this gene leads to a problem in learning and so context learning. So again, we repeated the electrophysiology, so like in SAT35, and what we found is that the electrophysiology really goes together with the behavior. And I also want to mention that all those experiments were all done in blind, because I'm always very afraid about the bias of the experimenter. So we are really revealing the genotype just after the analysis has been done. And so what we can see clearly is that in both cases, those animals of slices, in those hippocampus, we fail to induce this synaptic potentiation that we do expect when we stimulate with high frequency the Schaffer collateral of the hippocampus. So again, this is quite stable. The dynamic is slightly different, and I think it's important to notice this also in the context of what we see at the molecular level. Being H1L very fast, so being impaired from the very beginning, while KDM6B is showing the difference just starting from about 20 minutes after the delivery of the first of the stimulation. So going on the molecular side, so I'm going to go all the time back and forth basically between electrophysiology and molecular, because that's exactly what we are trying to do with this work, to try to understand the relationship. And so what we did is, as I mentioned, in this case, RNA sequencing and morphological analysis at the baseline. So what we wanted to understand is at the baseline already major difference in the transcripts eventually related to synapses, and or is that a difference in the synaptic morphology that could explain what we see. And the answer was actually no, and this was surprising. So what we are seeing here is that both in H1L and in KDM6B, we found very limited number of genes or transcripts that are differentially expressed between the mutant, so haploinsufficient animal, and the wild type. So in the H1L, we found roughly 50 genes, which are very, very few. And in KDM6B, we have about 10, which I rarely have observed, besides SAT35, where we observed exactly the same, whereas steady state, we really do not have major differences in gene expression. This, however, goes also along with what we found in the hippocampus, and I specify this because we think that there are also brain area differences where we do not find major differences at the level of the spine morphology and synapses number. So, good. So practically, again, because we are looking at the communication between the nucleus and the synapses during plasticity, we again wanted to go back and analyze the transcript of those animals during the plasticity, not just at the baseline. And so we repeated similarly to what I've shown to you for SAT35. What we did is, again, training those animals, obtaining their hippocampi 1 hour and 3 hours after the training, and then doing RNA extraction and library preparation and RNA sequencing. And we're starting by analyzing what we are observing in wild type. And so this time looking a little bit more about what is really the relevance of gene transcription during plasticity, this form of plasticity. And so what we see is that in the wild type, 1 hour after the training, we see a upregulation of gene expression, while there are very few or fewer genes that are downregulated. 3 hours after, the things kind of equalize a little bit, with actually a little bit more downregulated genes compared to the upregulated genes. I will go back to this in a second about what those genes are exactly. But what we observe instead in the mutant animals is that everything is much more attenuated. So those responses are kind of smaller in the sense that those two are H1L respectively 1 hour and 3 hours after the training, and those are KDM6B. And so you can see that the number of genes that are either up or down regulated during plasticity is much lower than the wild type. Now, of course, so here our first thought was that what happened here is that is simply synaptic plasticity is not induced in those animals, and so that this signal somehow does not reach the nucleus. But we think, in fact, this is not exactly the case, because when we look at early activated genes like CFOS, we see this is responding completely well. So in the same dynamics, same extent. So somehow the signal gets to the nucleus, but is somewhat attenuated and different. And so what are those genes? So again, looking at the wild type, what we found is that 1 hour after the training, we see a really very massive upregulation of genes that are regulating transcription, histone modification, and chromatin rearrangements. 3 hours after the training, most of those genes, they go down again. And in fact, we looked at most of them and really they show a biphasic kind of trend. On the contrary, synaptic genes show a different type of kinetics. And of course, I mean, here we are limited also on the number of time points that we did, right? Because it could well be that if we go more, so later, so we don't know, basically, you never know what happens all over the time. But what we see is that synaptic genes are just upregulated a little bit 1 hour after the training, but they are massively upregulated 3 hours after. What happens in the mutants is really nice, I think, is that the signature for histone modifier transcription-regulated, chromatin-related genes kinds of remain similar 3 hours after, but we are completely missing this upregulation of those genes 1 hour after. And in fact, what those trajectories often look like is like going downward all the time. On the other side, synaptic genes, they are completely missing. So we are really not inducing this second wave of gene transcription that are related to synaptic genes. So those 3 hours later, this upregulation of synaptic genes, really saying that eventually this dynamic so that there is between histone regulation and synaptic gene expression is relevant. Now, it's important to say that I'm not saying that those synaptic genes that we see upregulated are the ones that are in fact involved in that way of synaptic plasticity, but in fact that they may change synapses and the synaptic states for later on to be activated. So moving on, we were looking at, and again, I will go back to that as well, but moving on, we wanted to look at the homeostatic plasticity in mouse, so in vivo. I specify here we are doing in adult animals. And again, this is because what we are interested about is try to understand whether those genes are just relevant for synaptic functions during development or throughout life, because I think this will be an important information when we are thinking about treatment, right? If we need to maintain those treatment, how we have to, there are critical windows or not, and so on and so forth. And in fact, also the Hebbian plasticity part was done in adults, but also later, earlier on, to try to understand how this changes over development. So how are we studying synaptic homeostatic plasticity, and specifically in this case, upscaling in mouse is by injecting TTX in one eye. This kills basically all the projections of that eyes to the visual, so the contralateral visual cortex, but keeps intact the visual cortex, the projection to the visual cortex in the ipsilateral. This is really nice because practically what you have is in the same animal, you have both the control and the treatment. And so because we did in adult animals, we had to work a little bit with the protocol because this is typically done during this critical window of visual cortex development. But we show, in fact, that this works well. So those are the wild-type leader mates of the two genotypes that we were looking at. And we can really say that upon TTX injection, we have a increase of miniature excitatory postsynaptic currents amplitudes. That is what you expect when you upscale basically the synapses. So in this case, basically the neuron receive less activity. And so because of that, it's trying to increase the activity of the synapses to be in that sort of efficient range. And so that's what we see. So in the wild type, this is happening clearly. And what we see is that in both mutants, this form of plasticity is completely lacking. We still didn't do the other way around, so the downscaling is something that eventually we will have to do. But what we did is then moving forward with a human-based model and ask whether this is something that is conserved also in human neuronal circuits. And so in order to do that, I mentioned that we generated stem cells with mutation in all those 4 different genes that we were interested at. And we had to work a little bit with the protocol, because normally those organoids, they are not, so we use a classical classical like Lancaster protocol, but because those organoids, they usually are not maturing very much electrophysiologically, we had to change a little bit of things. And so what we're doing is then, when those organoids are about 2 months old, we are first exposing them or not to TTX to induce, again, to reduce the neuronal activity, to induce synaptic upscaling. And then we are slicing, 2 days after, we are slicing those organoids and do single cell patch clamp. This was something, again, that was never done before, so it took us a little while. So the very first nice thing is that indeed, by exposing those organoids to TTX for 2 days, we are able to induce this form of homeostatic plasticity, so this upscaling. So this is very clear in controls. And the very nice part is here, where we see that this form of plasticity is completely lacking in all the genotypes we are looked at. So really, to me, showing the largest amount of functional convergence among different genes that I have ever seen so far. So again, we want to also understand how this form of plasticity, that of this phenotype, electrophysiology phenotype, relates to the molecular, potential molecular issues. So what we did, and here I'm showing just the data on the mouse cortex, what we did is, again, doing this TTX injection, and instead of doing the recording, electrophysiology recording, we obtained the visual cortex from the contralateral and ipsilateral side and performed both bulk RNA-seq and bulk ATAC-seq, because of course those are factors that they're changing the chromatin structure, so we were also interested in looking at the structure. And we did that again over time, so both at 24 hours after the TTX injection and 48 hours after. And this is, I have to say, this is extremely difficult data to kind of interpret, and we are still working on that. And in fact, we think that the only way to understand some parts is go in single cell, and that's what we are doing exactly right now. But I give you a few, go through the data that we have so far. And so what we observe is that, let me go a step back. So what we did is looking at the RNA sequencing and data sequencing data starting from baseline. So again, the question was, is at the baseline, are there differences in the synaptic states of those animals in the visual cortex? And the answer is that in this case, contrary to what we see in the hippocampus, we could detect many more genes that were already at baseline, were differentially expressed in both an H1L and KDM6B in the mutants at baseline in the visual cortex. So really indicating that already at baseline there is something. And we don't know why there is this difference between the hippocampus and the visual cortex, where in the hippocampus we do not see any difference in transcription at baseline, while in the visual cortex we do. And interestingly enough, those differences are all related to synaptic signaling genes. But what we also observe is that the directionality of those changes are opposite in the 2 different genotypes. And so basically, to make it easier to digest, what we think is that despite those genes, they are functioning converging at the synaptic level, that do not necessarily converge in the directionality. And also, you can see here the overlap of genes. So there are just a few genes that are overlapping, those synaptic genes that are overlapping between the 2 genotypes. They are not overlapping in terms of the exact genes. So what we think is that the functional convergence is there, But this doesn't mean that the functional convergence is at the single gene and goes in the same way, in the same type of regulation. And I think this is very important. When we look at the ATA-SIC, we also found a difference, and I think this can explain partially, in part, what we see. And namely is that at the chromatin level, the H1L gene has already huge difference at the baselines with many differentially accessible regions between the mutant and the wild-type animals. While in KDM6B, we could not see a single differentially accessible region. So we do explain, or we kind of interpret this data, but again, for this we really have to obtain both the ATAC and the RNA from the same cell at the single cell level to really go into the exact mechanism, and that's what we're doing now. But the way we interpret here is that eventually those changes that we see at the transcriptomic level in the H1L are a direct consequence of those ATAC chromatin rearrangements that we see. But what we are observing here at the transcriptomic level cannot be explained directly by the chromatin rearrangement, and maybe it's just representing the synaptic state of those in those samples. So it's a slightly different type of mechanism, we think. And this makes sense when we move on and look at how the transcripts of those cells change over time during plasticity. So what I'm showing here is just at the baseline. So what happens during plasticity? So first, importantly, so those are just the wild type. So it's the same data that I've shown to you, or similar data that I've shown to you before. So what we see is that in wild type, both at the RNA sequencing and ATAC sequencing level, 24 hours after the TTX injection of those animals, not a lot is really happening, probably because still those nerves are still degenerating, and so the visual cortex still receive good inputs. And about 48 hours after, we see a really massive regulation of gene expression and also of chromatin remodeling. Interestingly, this goes in that, so has a slightly different directionality as compared to the Hebbian plasticity, whereby here we are seeing much more downregulation of gene expression, which kind of, again, we interpret in a way that sort of, in this case, gene expression is needed sort of to put a brake to the process that of homeostatic plasticity that is initially regulated by proteins that are already there. So basically by more translational dependent rather than transcriptional dependent. What happens in the mutants is that in the H1L mutant, where we had already those huge differences at the chromatin level, we did not see anything happen during plasticity. While in the KDM6 model, where we have no difference at the chromatin level before, we really have huge differences. Sorry, here is cut, but it's practically, in the ATA-SIC we see the same thing. So when we looked at the genes that are deregulated, so either at the transcriptomics or at the chromatin level in the KDM6 mutant, what we found is that the KDM6B mutants, they show a very much upregulation, so this is just the 48 hours time point, an upregulation of genes related to histone modification and chromatin modification, and a downregulation of synaptic genes, that, however, is a bit sort of not that strong. Interestingly enough, those factors that are activated here in KDM6B, they are not activated in the wild type. So what we asked is whether those factors that now are activated, so and specific transcription factors that are activated in KDM6B are somehow directly related to the expression of the synaptic genes that are differentially expressed in KDM6B. So practically what we did is creating gene interaction networks where we use the transcription factors differentially regulated in KDM6B with the synaptic genes differentially regulated in KDM6B, and ask whether those were more connected by the shunt. So in a way, it's like, are those transcription factors directly related to the synaptic changes that we see? And this is the answer is yes. So indeed, they are more connected than by chance, and also they are in fact also enriched for AAC-relevant genes. So really indicating that the changes that we see at the chromatin level are eventually directly related to the synaptic difference that we see, although we are not really saying what exactly the mechanisms are and what are the dynamics completely. So I have just a couple of more slides where I would like to show one part that it's really dear to us, is like how we can go about. So now that we know eventually that indeed those impact the role of those, so the synaptic plasticity during adulthood, can we do something for that? So can we go back to that concept of reversibility? And so I showed to you is that we also generate a number of stop animal models where you can induce, basically those animals are born as knockout animals. By tamoxifen injection, you could revert the expression of the one allele, and so you can convert them to wild-type animals. And I should mention that not all stop models work equally well. In fact, it's quite tricky. But I'm going to show the example of KDM6B that really is probably one of the best that we have in our hands so far. So what we're doing here is crossing the KDM, so the KDM6 plus stop animal. So the stop basically is like a minus allele. But the stop in this case is fluxed with the CRE animals, where we can then obtain those transgenic animals where we inject tamoxifen over the course of about a week for several times to induce the removal of the stop cassette to reinduce the expression of the KDM6B gene. And importantly, we do this when animals are already 4 weeks old. So again, what we want to ask is how this synaptic plasticity defect we see is a function of developmental issues or a function of KDM6 function later on. So we can show that we can indeed, so the stop animals that are here, that are just injected with vehicle, they show about 50% of the amount of KDM6 protein, so like we would expect, because they have one allele with a stop cassette, and we can show that we can not completely, but eventually revert partially the expression of KDM6B upon tamoxifen injection. And so then we did then afterwards is to do the electrophysiology that for us was very strong signature of those genes. And so what you're seeing here is like the two wild types, so the vehicle injected and tamoxifen injected wild type, where you can see really this nice long-term potentiation. Here you can see the vehicle injection and tamoxifen injection and KDM6 animals, whereby you see that those one are the vehicle injected, where you do not see these LTP inductions, so similar to what we see. In fact, we can also overlap with the heterozygous animal model. And here instead we have the animals upon reinduction or rescue of gene expression, so really showing that indeed this is not just a function of development. Finally, and this is really the last basically 2 slides, we wanted to leverage on the fact that we have this organoid system to try to eventually screen or do mini screens for drugs that eventually we could apply to rescue those phenotypes. And so here we could not really do a large screen. We're still doing patch clamping, so it's a low throughput. And so we had to do a sort of an educated guess of the drugs that we could use. So we went through the pathways of what we know that, for instance, H1L and KDM6B are doing. And we selected a couple of different drugs. I'm showing the data for one that show interesting patterns. So we selected this 1999 based on what basically this is an inhibitor of this PRC2 complex that is an antagonist of KDM6B. And what we show is indeed that, so we repeated basically the TTX induction, so homeostatic plasticity. And so what we show is indeed that this drug is able to specifically change or rescue plasticity in KDM6B, but not in H1L. So indicated by that, by using this approach with organoids, we could identify drugs that are specific to rescue. So it's not like the one drug for all, and I do not believe this will happen, but eventually that is able to rescue specifically the phenotype of KDM6B mutant animals or organoids. So with this, I really conclude. Basically, in summary, what I've shown to you is that we should also, as neuroscientists, focus not just what happens on neuronal activity or at the synaptic level, but we should look much more at what happens also in the nucleus during not just development, but also later on during life. And so that by analyzing a number of genes that are relevant to epigenetic and chromatin modification, we eventually found that mutations in those genes impact significantly synaptic plasticity, and we think that they do so by changing the response or the transcriptional response during synaptic transcription. And eventually, this is something I didn't tell you, but we are working on the hypothesis, that's why we need a single cell, that this happens because the system becomes much more noisy. And so in a way, the single genes that are changing they are not relevant, but what happens is that they're not fine-tuned anymore responses, but they're kind of becoming much less tuned with what happens. And so with this, I really want to thank the people. This is my lab. It's a fantastic, amazing group. And so Bernadette is the hero of the unpublished work that I've shown to you together with Christoph and Jessica. And so with the help of everyone else, as well as some other collaborators. So, and thank you for your attention.
So Gaia, amazing talk, really. It's the second time I've seen it, and it gets even better the second time. It's like watching a Star Wars movie or something. So when you present this to a room full of geneticists, human geneticists, huge light bulbs, red flags, and neon signs go off in our heads because it's been...
That's what they want.
It's been clear for years now that mirror phenotypes of different genetic disorders, and in some cases, the same gene, gain of function versus loss of function, producing a perfectly mirror phenotype, of both of which fall underneath the autism umbrella, has been kind of like a classic theme in human genetics. A theme that somehow doesn't ever seem to filter into the audience of neuroscientists, right? Where they're always looking for commonalities among all the autism genes, and the fact that they don't see them makes them worried, right? But that's actually exactly what's going on in autism, right?
That's right.
So you have the perfect example where you have two animal models that have exact opposite effects on LTP, exact opposite effects on conditioned fear learning, and then you start getting into the transcriptional effects, and you can connect transcriptional regulation to synaptic regulation. But obviously, as you get down those rabbit holes, we kind of lose sight of that original idea that you really have kind of these mirror opposites. So do you have a way do you have kind of a simple model to kind of describe, what's going on at the transcriptional level, and how are they going in different directions in these 4 different animal models that produce, and how are they affecting genetic regulation, synaptic proteins, and that produce these mirror opposite LTP and conditioned fear effects?
Yeah, thank you. This is exactly. I mean, and it's not by chance that I'm doing this work coming again from a lab where we did a lot of genetics and having some background in neuroscience there. So. The model. This is exactly where I am, and that is why I'm so. I really want to get this single cell, because I think we cannot get a real model until we really get into the mechanism precisely. And so I have tried to do this with the data I have in hands now for over 3 months and make it we want a simple model. And it's not simple. And one reason why it's not simple, that even if we look at 2 different brain regions, the hippocampus and visual cortex, we see slightly different things because we're looking, so even different forms of synaptic plasticity, they have different dynamics. So that's, so what I said at the end, that I think the one way to explain our data with a simple model is really a matter of noise to signal ratio is really what I think is happening. So it's about all noise to signal ratio. But why going in one direction or in another direction? This for me is not yet clear. Or in part it is clear depending on in which direction is going the expression of certain genes, that somehow if you are exaggerating the expression of certain genes, you may make those synapses just more. Still, they are not tuned, and that's why at the behavioral level, I think then they look very similar. But when you look at the electrophysiology level, they look different. So the transcriptomic level, they look different. So this, I think, really would explain the convergence at the behavioral level, and then start kind of making it subgroups at the molecular and the electrophysiology. So that's the way we are working on. But for that, I really need that data set, which should come in a couple of weeks.
Hi, Gaia.
Hi.
Fantastic work. You did so much experiments, so many mouse models, yes. And I was really fascinated, with your contact dependence data. So we all in autism research know that when you make haploinsufficient mouse model and look at the transcriptomic changes, they're always not that great.
Exactly.
And a lot of homozygous models, they are embryonically lethal, and we can study those, and then they're not relevant, right, to the human phenotype. So you really need to look at the haploinsufficient models. So my question is that, maybe do you think we all should be looking at the context-dependent, gene expression changes, proteomic changes, whatnot, and how do we pick the behavior? In your, model, it's fear conditioning, but there are so many different context-dependent, behaviors we can look at. What's your thoughts, in general, for autism research, looking at the, this haploinsufficient mouse models? How can we advance?
So thank you. This is an excellent question. And indeed, for instance, we are doing now the same thing we're doing in social behavior, right? Because this is something a different type of context. There's not but I think this is a very relevant question. And the answer is that so far what I don't know is whether a baseline, we cannot capture a difference, because in this case we are doing a bulk, and so basically we are not synchronized. So what my thinking is that it could be that what we are doing with the context or with the behavior is just sort of synchronize an amount of cells on the same response, and so we are able to capture something. And so while when we're doing just in a steady state, a baseline, maybe cells are all doing different things, so we're not really looking at the one precise thing. And so in order to answer to that, basically there are two possibilities. It could be that at baseline we're able to find differences, but just if we go in single cell populations. And so that's, again, is what we are doing right now, because this really will help us, is how much is context dependent, and how much is already a baseline, but because of the heterogeneity of cells, we are not able to capture. But on the other side, I pretty much believe that is also the other way around, that there is also something that is during, that we have to kind of play a little bit with those different situations to be able to capture different transcriptional responses.
Hi, great talk. One quick question. So I noticed that you also look into transcription factors along with histone modifiers. And for transcription factors, as they physically bind to the chromatin, and you're interested in the genomic differences among cells, have you ever done or considered doing a high-C sequencing to see how these transcription factors actually change and remodel the chromatin?
Yes, absolutely. This is on our to-do list that is becoming longer and longer and more expensive and more expensive. So it was nice to hear about the grants you have here. Just should go back with some of those to my institute as well. So one problem with IC is that tissue you start with at the beginning And I do not think that this is going to be feasible with the visual cortex or the hippocampus, because again, it's a lot of heterogeneous cell types, and so it's very difficult. One way we could do this, and we're thinking about do this, is with differentiated, so pre-differentiated cells, where we are, again, have a more homogeneous population, and then we could ask that question. A way to overcome this is that we could start doing, like, again, ChIP-seq and go more in that direction. But we are, of course, thinking about this, but we don't think this is the perfect model yet to do that. Thanks.
Evan, would you like to take some online?
Yeah, there's a simple one actually relevant to the haploinsufficiency question. The questioner wanted to know whether SETD5 knockout is lethal.
It is. All those, also as Lily mentioned, all those animals are lethal when in homozygosity. So we never work with that. Yeah.
Great.
Oh.
Hey, thanks for your talk. I couldn't help but notice towards the end, I never realized that some of these, I work in Alzheimer's disease, I never realized that some of these transcription factors are related to the PRC2 complex. And this is the complex that we are beginning to lean more on as a potential mechanism involved in Alzheimer's disease. Yeah. And interestingly, we see similar things. Some of the mutations in familial Alzheimer's disease result in opposite gene expression profiles. And so one of the ways we're trying to reconcile this opposing effect that we didn't expect to see is, well, generating time series data. Maybe the different mutations result in, an early activation of something that later leads to down activation of that same process. So we're reconciling it with time series data, or attempting to, and given all the similarities, I'm wondering what your guys' approach is, and in the future, how you're looking to generate, a schematic that reconciles that difference.
Yeah, I think this is very important, and we actually first thought about this when we were working on SETD5, because I mentioned that SETD5 is interacting with the HDAC3. And HDAC3 inhibitors have been proposed by, for instance, Liu Tsai at MIT, that could be used for treating Alzheimer. And so what we think here, and interestingly, if you're looking, and so we were screening the literature, looking at HDAC3 knockout mouse models, and the HDAC3 knockout mouse models have actually the same phenotype at the behavioral level in the context field conditioning as SETD5, so really matches that. And so this makes me think that indeed HDAC3 inhibitors, they're very good in eventually increasing cognitive abilities because of this learning thing that we also see in SETD5. So it's an increase of learning in the context of field conditioning. But what are the sort of the effect in the long term could be completely different, right? Because eventually this could lead to a decreased flexibility. So I'm not answering directly. So to your question, I'm just saying, like, we're aware about this, and I mean, on one side, of course, it's tempted to say, there is all this part that say how Alzheimer is really just a later time disorder, or how much is starting earlier on. So we have to start to put all this together. But I don't think I have yet a system to say, how to go about all those different genes and up and down and so on. This is exactly what also Jonathan was referring to. It's extremely difficult to find a one-fit-all rule.
Yeah, great talk. So just a quick question. Do you think the genetic background also plays in what you see, especially for the differences you see from each of the different genes?
Yes, I think one of the next steps will be interesting to look to generate cerebral organoids from patients. We do have cells in some cases, and to repeat all of this to understand what type of effect this will be really an important step to do it. Yeah.
Okay, maybe we'll finish up with one last question online. This questioner wanted to know, you clearly have isolated a lot of the defects to synaptic plasticity in looking at the various genes, have you been able to yet figure out what aspect of synaptic plasticity is it vesicle release, vesicle uptake, spine formation, that kind of thing?
Yes. So that's a very also interesting question. So and goes back to something that I briefly mentioned. So what do we see when we look at the enrichment analysis of, so GO-term analysis of synaptic genes, is that what we get back in the wild type are very specific terms. For instance, synaptic vesicle release and also exocytosis, but a number of other things, so postsynaptic density, so very, very, very specific. And what happens in the mutant is everything becomes broader. So that's why I mention is that what we think is that the system is not precise anymore. So it's not even about what type of genes, but it's like that everything become kind of more noisy. And so yes, we get still synaptic genes in some cases, but we don't get anymore those very specific terms. So in some cases it's synaptic vesicle release, but we have also other terms. Yeah, very proper question.
Okay. No other question? There's no other questions on Zoom. Any other questions in the room?
Great. So let's thank Gaia again for the fantastic talk.
Thank you.